Sorry about that. These are then called charged transfer RNA. And this is the amino acyl and the RNA, transfer RNA. So once it is released from the enzyme, it can deliver the amino acid to the appropriate place. The pairing of the transfer RNA anticodon with the appropriate messenger RNA codon helps to ensure that it's very, very accurate. In bacteria, you have 45 or more RNAs. Some must be able to bind to more than one codons to give versatility. This is possible because the third nucleotide base has more relaxed pairing. It's what we call flexible, or it has a wobble. Ribosomes facilitate the specific coupling of transfer RNA anticodons with messenger RNA codons. And this is happening, during translation. They are made up of large and small subunits, including proteins and one or more ribosomal RNA. In eukaryotes, the subunits for the ribosomes are made in the nucleolus. The ribosomal RNA genes are transcribed. The RNA is processed or assembled with proteins out of the cytoplasm, and the completed subunits are exported to the cytoplasm using the nuclear pores. Now, in ribosomes, both bacteria and eukaryotes have a small and a large subunit assembled only when they are attached to messenger RNA. A third of the ribosome is proteins. Okay? In bacteria, you have 3 ribosomal RNA molecules. In eukaryotes, you have 4. The ribosomal RNA is the most abundant type of cellular RNA. There's thousands and thousands of ribosomes. Eukaryotic ribosomes are larger. They're slightly different in their molecular composition. And this is medically significant because we have been able to create antibiotics like tetracycline and streptomycin that can specifically target the size of the bacterial ribosome and not the eukaryotic. Each ribosome has a binding site for messenger RNA and 3 binding sites for transfer RNA. The P site, which stands for peptidyl transfer RNA binding site, holds the transfer RNA carrying the growing polypeptide chain. The A site, the amino acyl transfer RNA binding site, holds the transfer RNA carrying the next amino acid that will be added to the chain. The E site, the exit site, holds the discharge transfer RNAs as they are preparing to leave the ribosome after they have deposited their amino acid. New amino acids are added to the carboxyl end of the growing polypeptide. The ribosome holds them close and catalyzes the formation of a peptide bond between the existing chain and new amino acids that are being added. As the polypeptide becomes longer, it passes through an exit tunnel in the large subunit, and when it's released, it exits through that tunnel. Ribosomal RNAs are responsible for the structure and the function of ribosomes. Proteins support shape changes of ribosomal RNA as it carries out catalysis during translation. The ribosome has a large ribozyme. Ribosomal RNA is the main constituent of the A and P sites and of the interface between the 2 subunits. This acts as a catalyst for peptide bond formation. Translation occurs in 3 stages. All require additional protein factors. Some steps of initiation and elongation require energy, and in order to do this, we're going to do the hydrolysis of guanosine triphosphate, GTP, which is very similar to ATP. Bacterial and eukaryotes both use the start codon AUG to start translation, and this establishes the codon reading frame. In other words, the group of 3 bases that are going to determine what is the codon. The small ribosomal subunit binds to messenger RNA and a specific initiator transfer RNA, and it carries the amino acid methionine. In bacteria, the small subunit can bind in either order. It binds messenger RNA at a specific RNA sequence just upstream of the AUG start codon. In eukaryotes, the small subunit is already bound to initiator transfer RNA and then binds to the 5' cap of the messenger RNA. And then it moves downstream along the messenger RNA until it reaches the start codon. And at that start codon, you have an initiator transfer RNA that will hydrogen bond to the AUG start codon. This will then be followed by the attachment of the large ribosomal subunit, which completes what we call the translation initiation complex. Proteins called initiation factors are required to bring all of these components together. At the completion of the initiation process, initiator transfer RNA sits in the P site of the ribosome. There's a vacant A site that's ready for the next aminoacyl transfer RNA. The polypeptide is synthesized in one direction from the initial methionine at the amino end, the N terminus. Excuse me. And this is toward the final amino acid at the carboxyl end, which is called the C terminus. And then this will be followed by elongation as the process continues.